Hello, my name is Ryan Benz, and I'm the Director of Data Science at Seer. Today, I'm going to present a poster of some recent work we performed titled Deep Plasma Proteomics at Scale with Proteograph Product Suite, a performance evaluation with label-free and TMT multiplexing methods. As you may know, deep proteomic experiments can be challenging, particularly for complex sample types such as human plasma. While deep proteomics workflows exist, there are typically several challenges associated with them. For example, there can be many steps in the workflows that are complex and time-consuming to execute. Furthermore, these workflows usually require significant domain expertise, making them less accessible to the general scientific community. At Seer, we are addressing this problem using the Proteograph, which utilizes nanoparticle technology to dig deeper into the proteome, along with sample prep automation to enable deep and reproducible experiments that are also high throughput. At a high level, the Proteograph workflow starts with a set of samples, such as human plasma, that are placed on the Proteograph automation instrument along with our nanoparticles and other reagents to automate the entire sample prep workflow. These samples are first incubated with the nanoparticles to produce protein coronas formed by the unique interactions between the nanoparticle surfaces and the properties of the captured proteins. These protein coronas are further processed, and at the end of the workflow, a 96-well plate of digested peptides is ready for LC-MS analysis. One question we wanted to understand is how reproducible the Proteograph workflow is. In the label-free DDA experiment shown here, we processed a pooled plasma sample using two different Proteograph instruments, each on 2 separate days. We assessed many reproducibility metrics, but here I'm highlighting the protein group intensity CVs. Across the different nanoparticles, Proteograph instruments, and prep days, we found median CVs typically around 20% or even lower. This shows that we can get highly reproducible data from the Proteograph with standard label-free workflows. In another experiment, we wanted to see how well the Proteograph could be combined with labeling and multiplexing methods such as TMT. In this experiment, we processed a pooled plasma sample in the same way as previously described, but at the end of the process, we labeled the samples with TMT Pro 16-plex reagents and processed with a standard TMT workflow. Again, we found the data to be highly reproducible, and in this case, we found median protein group intensity CVs below 10%. Furthermore, if we map these proteins to the Human Plasma Proteome Project database, we see that we detect proteins across approximately 9 orders of magnitude, showing that we can achieve both reproducible and deep proteomics measurements. To summarize, we found that the Proteograph platform can be used in conjunction with standard proteomics workflows such as label-free and TMT methods to produce highly reproducible and deep proteomics data. Particularly for researchers who are looking to run larger studies to gain more biological insight and power, the Proteograph provides the ability to run these experiments that would be otherwise impractical using other deep proteomics workflows. Thank you very much.